Imagine yourself at the top of the Burj Khalifa when you jump off the roof. But this is not the real Burj Khalifa. This is a scale model about 10 feet high, because you know that jumping off the real Burj Khalifa would be fatal. You sustain the fall with some minor injuries and bruises. You see that your very own skin cells are damaged. This is where the process of DNA replication must come into place, as each skin cell must regenerate with its own copy of DNA. This presentation will focus on leading strands and RNA primers. In the process of DNA replication, the protein helicase breaks bonds between the nucleotides which hold the double-stranded DNA together, splitting the DNA into two strands. This is where the process of DNA replication occurs. By creating a new strand that pairs with the template strand, you get two copies of the DNA. To actually create the new strand, two important proteins must be used: primase and DNA polymerase. DNA polymerase 3 is responsible for placing the new strands of DNA. However, the protein can't start from scratch. Instead, it needs a 3' hydroxyl to synthesize from. Because of this, the enzyme primase first synthesizes a fragment of RNA called primer, about 5 to 12 bases long, by pairing complementary bases with the template strand. The primer ends with a hydroxyl group, which gives DNA polymerase 3 a starting point to attach to and to begin synthesizing DNA. DNA polymerase only works in the 5' to 3' direction, which is why one strand, the leading strand, is synthesized continuously, while the other, the lagging strand, is built in fragments since it runs in the opposite anti-parallel direction. Once DNA polymerase 3 has begun synthesizing the new DNA strand, DNA polymerase 1 is introduced and replaces the RNA primer with DNA. This effectively creates one new continuous strand of DNA, which when paired with the old strand, creates a completely duplicated piece of DNA. Thank you for watching.